Hi, I'm Dr. Jasmine from the Department of Biotechnology, Bishop Heber College, here to discuss about the role of telomeres in cancer. What is the role of telomeres in cancer? We first need to know about the difference between normal cells and tumor cells. Normal cells divide for a limited number of times, after which it undergoes a period of cell death, which is called as cellular senescence. So they have limited growth and they follow apoptosis, which is programmed cell death, whereas tumor cells usually have the ability to proliferate indefinitely. That's why they are called as immortal. There is no death. And the picture shows the normal cells towards the right and the left, and towards the center we can see the tumor cells, which grows in an undifferentiated mass. Aging and cell division. So there is evidence that cellular aging is dependent on cell division. As the cell divides, then after a period of time, it undergoes death. So the total cellular lifespan is measured by the number of cellular generations. And what are telomeres? The tips of the chromosomes are called as telomeres, and they play an important role in aging. Let's see the role of telomeres in aging and the role of telomeres in cancer. It is now evident, or rather proved, that the loss of telomeric ends of chromosome is important in human cell aging. So loss of telomere means more aging. So telomeres are the ends or the tips of the chromosome, and as the cell divides, as shown in the figure, you can see the red portion, which is the telomere, and as the cell grows and divides, there's depletion in the telomere, and towards the end there are n number of cells where the telomere is completely depleted. Therefore, you can see the role of telomere in aging. What is telomere? Telomere is a region of repetitive nucleotide sequences at each end of the chromatid, as shown in the figure. What is its function? It protects the ends of the chromosome from damage, deterioration, or from fusion with the neighboring chromosomes. So it forms like a cap at the ends of the chromosome. So this is a beautiful photograph showing the telomeres in the chromosomes. Its name is derived from Greek telos meaning end and meros part. For vertebrates, the sequence of nucleotides is TTAGGG, and this is a repetitive sequence approximately about 2,500 times in humans. Isn't it amazing? So this repetitive sequence is a significant part of the telomere. What is telomerase? This is an enzyme that replenishes the telomere. It is found in the eukaryotes, because you know the prokaryotes chromosomes are mostly circular with few exceptions. So this telomerase is a ribonucleoprotein complex, and it adds the telomere group, that is TTAGGG repeats, onto the telomeres. This enzyme telomerase helps to stabilize the telomere length. In other terms, this telomerase helps to maintain the length of the telomere. As the cell grows and divides, the telomere shortens and later it depletes, resulting in cell death, whereas in cancer cells, these telomerase enzymes, they never get depleted, and this helps to replenish the telomere. As a result, the cell undergoes constant division without apoptosis. More telomerase activity has been found in human tumors, but not in the normal cells. So telomerase activity is now connected with tumors or cancers. So as shown here, telomerase was discovered in 1984 by Elizabeth Blackburn and Carol Grader. So this is a pictorial representation showing the enzyme telomerase, and this has the RNA template along with the nucleotides. So the entire complex is seen to replenish the telomere. What is the mechanism of the normal cells? So as shown in the figure, telomere shortening occurs in humans. This induces replicative senescence, which is cell death, and later blocks cell division, prevents genomic instability, which means it helps in the maintenance of stability of the genes, and limits the number of cell divisions. So in other words, as the cell grows and divides, there is shortening of the telomere, and this results in cell death after a certain number of divisions. This is a mechanism found in the normal cells. But this is not seen in the cancer cells. It's a reversible mechanism where telomere shortening does not occur, as shown in this particular slide. So the mechanism of telomere in cancer cells. Telomere shortening happens as the cell divides, but here it induces telomerase production. So this telomerase enzyme, it will replenish the telomere. Therefore, there's increased replication, immortality, which means uncontrolled cell division, leading to the instability of the genes and development of cancer. So what is the major difference? The major difference is the production of telomerase, which replenishes the telomere, and as the telomeres are replenished, the cells are immortal. Telomerase is a target. Now this telomerase is now chosen as a marker for cancer diagnosis. So there is a limit of the telomerase, and when there is excess of telomerase, it means that the person suffers from cancer. So now this is used as a target for the development of novel anti-cancer therapeutic agents, and researchers focus on this telomerase to prepare and design drugs. Detection of telomerase in cancer diagnostics. Once we are able to detect the alarming increased quantity of telomerase, we can always relate it with cancer. Most human cancers have short telomeres, but it is replenished because those cells express high levels of telomerase, and hence telomerase is considered as a selective biomarker for screening. It is useful for early cancer detection. As shown in the figure, the genome or the DNA or the chromosomes of prokaryotes are circular, whereas in eukaryotes it is linear. Since they are circular, they are not in need of telomeres. Hence, telomeres are seen only in eukaryotes. But there are certain exceptions like Streptomyces and Borrelia, which have linear chromosomes and therefore they possess telomeres. Apart from Streptomyces and Borrelia, all other prokaryotes, chromosomes are circular and they are devoid of telomeres. So the major function of telomere is to protect the tips of the chromosomes. And the cell has the enzyme telomerase, which helps in replenishing the cap, that is a telomere. And in most of the multicellular eukaryotic organisms, telomerase is active only in the germinal cells. Even in the stem cells, like embryonic stem cells and certain other white blood cells. Now I hope you will be aware of the immortal property of the stem cells, and you can relate it with the presence of telomerase. So this is a beautiful picture, and observational studies have shown that shortened telomeres in many cancers have been replenished by the telomerase enzyme. Diagnosis. A rapid shortening followed by stabilization of 3 to 4 years before cancer is diagnosed. And this could yield a new biomarker to predict cancer development with blood test. So this presence of telomerase is a very good indication of the development of cancer. So you can see high quantity of telomerase in cancer cells as well as in stem cells. Scientists have now been trying to understand how these telomeres are considered as a marker, and people who are affected with cancer, they have high quantity of this telomerase enzyme. See, telomeres shorten every time as a cell divides, as we have seen earlier, and older we are, the more times the body, the cells has divided, and it shortens the telomeres as shown in the figure towards the right. So the left shows the presence of telomeres and the right showing the shortening of telomeres, which is associated with aging. So as a cell, this is a very interesting figure. As the cell begins to become cancerous, it divides very rapidly and the telomeres become very short and the cell dies. Whereas in cancer cells and stem cells, telomerase comes into function and thereby it replenishes the telomere and thus prevents cell death. How do we measure telomeres? Studies have found shortened telomeres in many cancers as shown here: pancreatic, bone, prostate, bladder, lung, kidney, head and neck. And measuring telomerase enzyme is a new way to detect cancer and also the staging of cancer. So greater the quantity of telomerase, we know that the cancer is in the advanced stage. So if scientists can learn how to stop or block this telomerase enzyme, then I think that will be a very good breakthrough in fighting cancer cells. So blocking telomerase has now become the aim of researchers. In one experiment, researchers blocked telomerase activity in few cancer cells growing in the laboratory, and to their surprise, they found the tumor cells to die. So tumor cells could be targeted by blocking telomerase. So blocking telomerase has a lot of promises, but still it comes with risks. What are the risks? So they found that the risks involved with blocking telomerase included fertility problems, wound healing problem, production of blood cells and immune system cells. So problem with all these. So impairment was related with blocking telomerase. Knockout mouse. Studies using knockout mouse where genes are removed have demonstrated the role of telomerase in production of tumorigenesis. So the relationship of telomerase in promotion of tumorigenesis was proved using knockout mouse. What is the future research? Researchers are now breaking their heads to find out new, or rather novel chemicals or natural compounds which could block the telomerase activity without impairing other organs or without having any side effects. So in future, I hope that agents to block telomerase will become a very successful model to control cancer. Thank you.